Okay, there it goes. Okay, well, first of all, thanks for the invitation to speak to you all. When Fred asked me to do this, he had mentioned that you might have a lot of students, so I hope I didn't make the first part of this like too introductory. Let me see, how do I advance my, there it is. So this is kind of who I am. I've been a research entomologist with the Systematic Entomology Lab for going on 40 years. And my main responsibilities are research on plant feeding flies, mainly Tephritidae. And I'm also one of the curators of the USNM fly collection, which has about 6 million specimens in it. And then I also do identifications of specimens, mainly for USDA APHIS. And to start off, first of all, what I'm talking about tonight are not Drexel, which is another family that are commonly called fruit flies or pomace or vinegar flies. Drexel have a really diverse biology, but they're generally not phytophagous. Drosophila melanogaster is one of the most important model organisms in biology. I'm sure you all know about it, but we're not talking about them. So I'm talking about what are often called true fruit flies. And these are extremely important to agriculture, which is why I have a job. At least 50 species are considered major pests of a whole range of fruits and vegetables. Over 200 species have been reared at one time or another from economically important plants. Some Tephritidae are really broad generalists, like the Mediterranean fruit fly and the Oriental fruit fly. Each have hundreds of reported host plants, and lots of others are potential pests. And the pests cause direct losses in the billions of dollars, and they also prevent development of fruit industries. Because if you have some of these pests, you really can't export without a lot of treatment. And most of the pest Tephritidae are exotic to the US, and preventing their introduction into the US is a high USDA priority. There's extensive trapping systems in various parts of the country, and it's fairly expensive, but establishment of a major pest would cost billions of dollars. Like if the medfly got into California permanent, for example, lots of Asian countries would immediately shut down the export of citrus and other fruits to those countries. So one of the best ways to keep the pests out is to know as much about them as we can, including their distributions and what hosts they can hit, and that's really key to the quarantine regulations. There are really sophisticated eradication methods for a lot of fruit flies, but they're very dependent on what species is involved. So knowing the taxonomy of the of a new pest that comes into a country is really critical. These are just some of the invasive species that have gotten into North America. There's 11 species that are exotic that we now have. I included Hawaii in this. As I mentioned, there are really good eradication methods, but they're very complicated and they are very species specific. So one of these is the sterile insect technique, which involves mass rearing huge numbers of flies, irradiating the males, and then releasing them to mate with wild females. But obviously, if you don't release the right species, that's not going to work. There's another technique called male annihilation. There's some Tephritidae that are really strongly attracted to certain lures. So you can put out baits that are laced with pesticide to get rid of those. But again, you need to know which species it is, so you use the right method. But The vast majority of Tephritidae actually don't have any direct economic impact, and there are some species that are beneficial and have been used as weed biocontrol agents. For example, there's 10 species introduced to North America and 12 to Australia or New Zealand. Most Tephritidae are phytophagous. Some Phytomyinae are subtropics, and at least one taxonicid is a parasitoid, and we think that probably that whole subfamily are parasitoids. Larvae of Euphranta taxonura prey on softfly larvae inside of the softfly's gall. It's kind of an oddball. So their biology is quite interesting. Here's another one that's interesting. There's a whole group of Tephritidae in the paleotropics that breed inside bamboo. And this one in particular has a unique strategy. It finds weevils which chew holes into the bamboo, and they wait around the weevils, and then after the weevil finishes ovipositing, they lay their eggs through the same hole. And they got to act pretty quick because bamboo grows so fast that the holes close up pretty rapidly. More about the biology. Adults generally don't feed except on liquids or particulates in the regurgitated saliva. You see here over on the left. But there are 3 genera of Blepharinorinae that have spines on their labella, and they can actually rasp leaves or flower tissue. But generally, it's the larval stages that do most of the feeding. Despite the common name of fruit flies, there's actually the majority of species feed on other plant tissues. The larvae can feed on fruits, flowers, flower heads of composites. Some are leaf or stem miners. Others induce and feed in galls on various parts of the plants. They're usually brightly colored. The wings usually have patterns, and it's used in courtship and other behaviors. There are more than 5,000 described species, but we know there's a lot more undescribed out there. And they're cosmopolitan, but the greatest diversity is in the tropics. Here's some species that have kind of unusual morphology. This is an Anastrepha with the female has a really long ovipositor. This is one of what are called the antler flies. They have these processes on the sides of their heads. There are some stalk-eyed Tephritidae. In some species, the males have modified setae on the fronds. Sometimes they're on these projections. The courtship and mating behavior is often very complex. The males generally produce pheromones to attract females. Some species lack. Males are sometimes territorial. They'll guard mating sites or oviposition sites. They sometimes will sort of joust with each other. Courtship usually involves various wing movements. Sometimes they dance around. Some species do what's called trophallaxis, where the male may pass the fluid to the female, and other males produce sort of like a froth mass. It's a nuptial gift. And females often mark fruits with pheromones to deter other females from ovipositing. So you could do a whole presentation just on fruit fly biology. It's quite interesting. As I mentioned, there's more than 5,000 species known. And there's a lot of undescribed species in the tropics. Just some examples. In the Molinocelia group, before I revised it, there were only 3 recognized species. Now there's 36. In Blepharinorinae, there's currently 53 species recognized, but Marty Condon's estimated there's more than 200. In Anastrepha, about a third of the species have been described just in the last 20 years or so. The distribution is cosmopolitan, but the greatest diversity is in the tropics. Okay, said that already. Here's a breakdown by region. As you can see, the paleotropics are the most diverse. The oriental and afrotropical regions have the most species. And where we are in the Nearctic is actually the least diverse. And so these are just the general goals of what I do. A lot of it is just very basic taxonomy. We're just trying to find out, what species are out there, how do we tell them apart, and we're trying to find, better, more reliable and faster methods for identification of the pest species. Try to provide identification tools, descriptions, illustrations, keys, etc. And then we want to know what's the correct name for each species. And we also try to investigate the phylogeny or the evolutionary relationships as a basis for the classification. Fieldwork with Tephritidae is really important, just for collecting material for study. I've reared a number of species where we hadn't seen them before, and so by looking for infested plants and rearing things out, you often find things you don't collect as adults. And then lastly, I try to compile biosystematic information in databases. There's so many insects out there, just to try to provide a single source for users for the names, host plants, and distributions for Tephritidae. I was mentioning again, fieldwork is really important. And so here's some examples of publications. This first example is from the Manual of Central American Diptera, which is a really basic identification guide. If you've got, so the manual includes chapters on all the families of flies that occur in Central America. So if you're trying to identify a fly from that area, this is one of the first places you would go. First to identify the family and then to genus. So I did the Tephritidae chapter for this, and it includes a diagnosis of the family, a key to all the genera from the neotropics, and then synonymies, what we know about the genera from, for each of those genera. I try as much as possible to do comprehensive work or revisions. So basically, you want to try to pull together all the specimens you may have or that you can borrow from other museums, and try to figure out what are all the species, how do you tell them apart. You want to provide a diagnosis, the correct name, key, illustrations, and here's some illustrations from this Melanocelia group. Tephritidae, body patterns are often important. Wing patterns are very important, and often there are differences in the genitalia. And then we're trying to reconstruct the evolutionary relationships as well. And like for morphology, that usually involves trying to compile all the characters into a matrix, looking at related flies to figure out what are the primitive and apomorphic characters, which are what generally indicate relationships. Then you plug it all into software to analyze that, and then we get these phylogenetic trees showing what's more closely related. And we're generally looking for what are called synapomorphies or shared derived characters. And sometimes it's really obvious, like the genus Pseudophorelia has a number of really distinctive, unique characters. For example, on the abdomen, it has this Richardiid-like seta on the basal segment of the abdomen. It has this vein, the base of cubital vein, setulose, which is not unique to Tephritids, but it's not common. In the male genitalia, other Tephritids generally have one or none of these lobes at the base, and Pseudophorilia has two of them. And then in the spermathecae, they're also unique. This is a typical looking Tephritid spermatheca. Sometimes they have little scale-like spines, but Pseudophorilia has these big sort of globules on it. So it's, very obviously a monophyletic group. I tried to put in some examples of, how we might distinguish closely related species morphologically. And generally what we're looking for are correlated gaps in morphological characters that might be indicative of reproductive isolation. So for example, in the USNM collection, there were specimens that had this kind of wing pattern, and I noticed that some of them were different. In some cases, this band was right along this vein, but in others there was a hyaline area. So when I looked at the genitalia, the ones that have this wing pattern have this kind of ovipositor tip. There's no serrations on it, but the ones that had this character also have this. So, the hypothesis there is that those are two different species. And here's another example from Blephara neura. In Bolivia, I was collecting one place and I got all these flies basically off the same plant in the same spot. And I originally thought they were all the same, but looking more carefully, the genitalia had basically three different forms, and those turned out to be correlated with the wing pattern, basically mainly with the size of the spot in this cell. And I can figure out how to do it. I was gonna give you a demo of the ID system we're working on. Do you see something different now?
Yep.
Okay, so for Anastrepha, which is one of the largest genera of Tephritidae, there's over 300 species currently. I've tried to develop a interactive key, because with that many species, like using like a traditional dichotomous key gets really, really difficult. So basically, you just you plug all the information into a matrix, and so here's all the species listed over here on this side, and then these are all the characters that you can use for identification. So you can pick any character you want, but I'll just pick this one at the top. You would be looking at your fly, and this is a part of the ovipositor, which is used by the female for laying eggs. So, let's say if the one you had is 4 millimeters long, you would hit that, and then you can see that really eliminated a lot of the possible species. And so you basically just keep doing this as you go step by step. You can pick any character you want. Let's say your specimen has this character, this marking on the hind part of the thorax, and that eliminated a whole bunch more. And it's really nice to have these systems because it pulls together all the information together. We also try to include images of the whole body, of the wing patterns. This is the ovipositor in the female. You can also see a full description, or just a diagnosis. You can also do things like, if you are not quite sure which species you have, you could select a couple of them, and then you hit this button, and then it'll give you a list of all the ways that these species differ from each other. So anyway, back to the presentation. And then I mentioned before, one of the other things I try to do is to compile all the information about Tephritidae names, host plants, and distribution. We put that information on it's the USDA Compendium of Fruit Fly Host Information, or coffee. So if you are looking for information on fruit flies, this would be a good source to use. Now the next two parts are actually two different presentations. I just kind of pulled them in here together. So one of them was from the Entomological Society of America meeting in November of 2021, and it's talking about a project that we're doing to try to determine the phylogeny within the Tephritidae. And that's Brian Wiegman and Keith Bayless are the main collaborators, but there's a whole bunch of other Tephritidae systematists and other people who are contributing material. So what I was trying to do in that presentation was sort of present an overview of what we know about Tephritidae phylogeny, and then show some of the preliminary results. Let's see, I'd probably skip some of this here. Molecular phylogenetic analysis on Tephritidae has been pretty limited. And we still have like a lot of questions. Like most of the phylogeny is currently based on morphology, but we still have a lot of questions about relationships among some of the higher taxa, and even with some of the other related families. Well, these are some of those previous studies that we mentioned. And so for this project, we were using two general methods, which both yield large amounts of DNA. They've been used to investigate relationships within various other fly families. And the two methods are transcriptomes and anchored hybrid enrichment. The vast majority is from the latter material because it's a lot easier to get samples. You don't need special preservation methods like you do to get transcriptomes. I'm not going to talk too much about the details. And I mentioned we have collaborators from all around the world who are contributing to this project. So far, well, this is a little bit dated, but of the 500 or so genera of Tephritidae, we have results for about 102. It's probably getting closer to 200 at this point, and we have representatives of most of the higher taxa, which is good. And this is just kind of a list of which taxa we have. I'll just skip through this because it's probably only of interest if you're especially interested in Tephritidae. And this was the preliminary results for the 152 taxa. We still do need a lot more taxon sampling to flesh it out better, but we feel like we're getting there. There's about 500 genera of Tephritidae and 5,000 species or so, and they're classified in 7 subfamilies and almost 40 tribes or subtribes. Tephritidae belong to the superfamily Tephritoidea, which includes 10 families. It's a pretty well recognized superfamily. There's still some questions about relationships among some of the families, and we're also trying to resolve those, but that's not the main focus for today. There's general consensus that the Platystomatidae, Pyrgodidae, and Tephritidae form a monophyletic group. They have a couple of morphological characters which indicate that, like particularly the male phallus. It's only in these 3 families that it's stored up in this pocket up here instead of just kind of coiled down at the base like in the more primitive families. And they also have generally clustered in molecular studies. The most important morphological characters are mainly the shape of the subcostal vein, which has a strong bend to it, and also the presence of frontal setae. And those are unique to Tephritids, except there's also present some Eulideids, which is a related family. But those aren't perfect characters. Like there's sometimes reversal. For example, in some tachynicines, the subcosta isn't as strongly bent. And then in some basal pergodids, like this one here, they also have a strong bend. We're especially interested in this genus Descoleia, which seems like it might be kind of a intermediate between pergodids and Tephritids. And we got some specimens recently, so I'm looking forward to seeing where they come out in the molecular study. And in the past, some molecular phylogenies have placed the tachynicinae and or Blepharonura closer to the Pergodidae, but the number of samples has been extremely limited. And as I mentioned, relationships among the Tephritid subfamilies are rather poorly resolved. There's a lot of nodes that are basically bushes based on the morphological studies. And there hasn't been much support for any of the branching in the molecular studies that have been done. So anyway, the preliminary results from our study, it does confirm the monophyly of the Platystomatidae, Tenostyle, Pergodidae, and Tephritidae. However, the Pergodids are actually nested within the Tephritidae. They're clustered with the Blepharonurae and the Phytalmyaenae. So, you might conclude from that Pergodids perhaps are just sort of derived Tephritids. We didn't, unfortunately, have 2 of the representatives of 2 of the subfamilies when this analysis was done, so we're not sure where they go yet. And then this clade seems to be the sister group for the remainder of the Tephritidae. And then one of the clades that Korniev, who's done a lot of the morphological analysis of Tephritid phylogeny, he felt that this was a well-supported clade, and that also is supported in this tree. So the Tachynicinae, it's sometimes been considered a separate family. Some of the genera kind of look like tachinids with Tephritid wings and genitalia. Some of them are very stout and bristled, but others look much like typical Tephritids. It's a very small group, which is one of the reasons it's been hard to get good samples of them. The biology is pretty known. The one species is a moth parasitoid. It's pretty clearly a monophyletic group based on morphology, but there hasn't been much molecular study of that group. This is the Blepharonurae. It's a bit more diverse. There's 65 species currently, but we know there's a whole lot more Blepharonura out there. Marty Condon's done a lot of work, really interesting work with Blepharonura. The group is pantropical, and at least 2 of the genera develop feed on cucurbits. And in our study, at least, the monophyly of at least 3 of the genera is really very strongly supported by morphology. But there haven't been any molecular studies that included more than 1 genus. Another subfamily is the Phytomyiinae. There's about 300 species. They're paleotropical in distribution. Some develop in fruits, but many are subtropics, although they tend to specialize on certain plants or tissues, like feeding on bamboo or on the cambium of recently fallen trees. There's some morphological support for their monophyly, but Korniev was unsure if some of the tribes really belong. Only 3 genera have been included in molecular studies, and they did cluster. And let's see, I'll skip through this, I think. And so in our study, we can't really evaluate the monophyly of the Blephara Norrinae yet. We only had 1 sample so far, although we have more samples in the next tree that'll come out. But the Phytomyiinae, we have 6 genera, and those all did come out as a monophyletic group. It's this green group up here. The next subfamily is the subfamily Decinae, which is one of the largest subfamilies. There's about 1,300 species. They're also mostly paleotropical. 2 of the tribes feed on fruits, and the third feed in stems of bamboo or grasses. There's a lot of pest species in this group. There's pretty strong morphological support for this group being a natural group, and also far all the molecular studies that have been done have also supported that relationship. I guess I was going to go through the tribes. There's the tribe Decinae, also is very strongly supported molecularly and morphologically. There's another tribe, the Tephritidae, that includes Ceratitis, which is includes the Mediterranean fruit fly. They've been considered monophyletic based on morphological evidence, and so far there's no sign from molecular studies that they're not monophyletic. Although there's some question about whether the Tephritidae and the Gastrozonini might be monophyletic. And then the Gastrozonini, based on morphology, there's no really obvious characters to support its monophyly. So in our analysis, the subfamily comes out as being monophyletic, as do each of the tribes, although we'd like to increase the number of genera we have in the sample size. And then yeah, it seems like the Tephritidae and the Gastrozonini are more closely related. The next subfamily is the Tripetinae. It's another very large subfamily. There's quite a few tribes, 9 tribes, and it's also widespread, and the biology is fairly diverse, although a lot of them are fruit feeders or leaf or stem miners in the Tripetinae. It's unclear if the subfamily is monophyletic. It very possibly might be a grade rather than a clade. Most of the tribes, especially the smaller ones, seem like they're monophyletic, but the relationships among them have been very unclear. Han and Rowe included representatives of 4 of them, but they did not cluster, and there's really pretty poor support for those parts of the tree. So it's kind of inconclusive. I was going to run through some of the different tribes, but I think maybe I should skip that for purposes of time. And our results so far show that most of the Tephritidae looks like it's monophyletic. It's this clade up here, but one tribe seems, the Adramini seems to be closer to the subfamily Tephritidae, but these others so far are clustering as a sister group to these other three taxa, and there's good support for that. The tribes Adramini and Tephritidae are each monophyletic in this tree, as are the bulk of the tribe Carpomyini. And other two tribes seem to cluster as well, which was there are two groups that Korniev hypothesized might be related. The next subfamily is the Zaceratinae, which we unfortunately didn't have a representative of yet. It's a very small subfamily, only 2 species, but it's been hypothesized to be the sister of the Tephritidae. And then the last subfamily is the Tephritidae. It's the largest subfamily with close to 2,000 species, 13 tribes. It mainly develops, like most species, develop in plant tissues of the family Asteraceae. There's a couple groups that hit other families. There's no real clear-cut morphological synaptomorphy for the group, but they're generally considered monophyletic, and they've been monophyletic in all molecular studies to date. Here's just some different examples, different genera. This is by far the most complicated subfamily. I won't go into all of the details. I tried to sort of color code the ones that we think from previous evidence are likely to be monophyletic. Orange ones we feel pretty strongly are. The green ones, probably they're monophyletic, but maybe a few taxa need to be changed around. And then these blue ones are pretty questionable whether they're monophyletic or not. And these are the preliminary results that we have. And generally the groups that are well supported from morphological evidence are also supported in the tree, which is a good sign. The two tribes that feed on non-Asteraceae, neither one of them comes out as monophyletic, but they are clustered together along with 2 genera of another tribe. So there seems to be some importance there of the host relationships. And then these 3 tribes look like they're highly polyphyletic, so they probably need to be completely reevaluated. So just as a conclusion, results from the study are rather preliminary so far, but we're encouraged. We're trying to get other people at the symposium to contribute samples. Now, I don't know if I'm going over time, Fred. I was going to conclude with some, this is a presentation about the genus Anastrepha.
Go right ahead, Alan, you're fine. Go right ahead.
Okay, I'll keep going. And so this was a presentation at a meeting of fruit fly workers last year. And again, this is a part of a big team. We have a large group of scientists from the US and various other countries who are contributing. Anastrepha is the largest genus of Tephritids in the neotropics. It's also the most economically important. There's major pests that hit lots of critical crops like citrus and mango and wide range of other things. And there's quite a number of other lesser pests or potential pests. The taxonomy currently relies largely upon characters of the adult female. The males and immatures of many species are difficult or impossible in some cases to distinguish. And the phylogenetic relationships have also been incompletely understood. So we had some money from USDA Farm Bill, and the purpose was to try to develop better diagnostic tools, including for the larvae, as well as more robust phylogeny. So we were trying to describe additional species. I showed you the demo of the Anastrepha interactive key, trying to expand that. Some of my colleagues have been working on the morphology of the larvae and also developing a identification key for them. We've done a lot of CO1 barcoding, which is useful for species identification. And we've also been trying to explore other markers and develop rapid assays for some of the pests. And then we were using the anchored hybrid enrichment method, like the same thing we used for the Tephritidae phylogeny, to develop a better overall phylogeny and also try to find different new diagnostic regions. So sampling was really a key component of it. We invested a lot of resources in collaborators in other countries. We identified good places to collect and provided funds for them to run the trapping surveys so we could get good material. And one thing I think it's often overlooked when people propose discover new methods and things, but if you don't have the samples to look at, those methods can be wonderful, but they don't really do you any good if you don't have the material to analyze. So we did extensive surveys in northern Brazil, in Ecuador, Peru, in Suriname, and then we did some shorter collecting trips to several other countries. And we used what are called multi-lure traps to collect the flies, and they preserve the DNA better than the traditional lures that have been used. And we also tried to rear material from fruits. And then we also got material from collaborating agricultural agencies in some other countries as well. These are our collecting sites. We tried to sample from across the range of the genus as best we could. And these are some of the results. We found more than 50 previously unrecognized species that have been described, and we think we have even more additional species that we think are new species that still need to be described. As I mentioned, we've been expanding the interactive key. These are just some of the new species that have been found. Gary Steck and Eric Rodriguez, colleagues in Florida, have been working on the larvae. They found a number of new characters, like on the underside of the mandible. Like they found that the surface is quite different. Some have grooves, some are basically flat, others have various bump-like structures on them. They've also found characters in the what are called the oral ridges and a number of other characters. We have a lot of new host data. We have a manuscript about ready to come out with new data for 40 species. A lot of them, they're the first known hosts for those species. I mentioned we've been trying to do CO1 barcoding. So we've developed a quite large sequences for 273 species. Let's see, I can move on. And then one of my colleagues has tried to do a lot of analyses of it to try to figure out, what's the best methods for using the data for diagnostic purposes. And what we found is the CO1 barcodes are significant to diagnose the majority of the species, about 75%. And most of the species that can't be diagnosed are in what's called the Phytophagous group, which includes a number of the pest species. They've been useful to confirm the identity of some of the larvae that were used in the studies I previously mentioned. Beyond that study, we have another batch of new data that's coming along. This is sort of a summary of the geographic coverage. As I mentioned, one of my colleagues, Raul Ruiz, who works for APHIS in Texas, he's trying to develop rapid assays for some of the main pest species. So one of them is almost ready for one of the cryptic species in the Anastrepha furticulus complex, and we're working on some others. In terms of the phylogeny, the most comprehensive study previously available was by Chimo Mengual at Alia. Chimo was a postdoc in our lab, but it was only based on 6 genes, and there was good support for a lot of the species groups have been recognized in Anastrepha, but relationships among them were really unclear. So the excuse me, the anchored hybrid enrichment tree, that method produces a ton of DNA data. So the one you see here was based on 470 specimens, 170 species, with at least 21 loci represented in most of the taxa. The results are still preliminary, but this phylogeny does resolve many of the uncertain relationships within the genus. It corroborates the inclusion and synonymy of the genus Toxotrypana, which is a rather derived looking Tephritidae, includes the papaya fruit fly, which was previously recognized as a separate genus, but it seems to fit. It's nested well within Anastrepha, as you can see this dark green area. This tree also corroborates certain clades that have strong morphological support, like there's 2 species groups here, the Dasyphoremus and Dentata groups that have a lot of morphological support, and they come out together. Most of the species groups are confirmed as monophyletic. I mentioned the Phytophagous group before. That's this whole big clade here. it's monophyletic. Another group, the Striata group, is monophyletic. I tried to sort of color code some of these and point with arrows. So like some of the species groups, like the Pseudoparallela group, are mostly monophyletic, but then there's like 2 species that apparently don't belong. They don't come out together with the rest. Some of the groups are monophyletic or close to it. So, you know, we're pretty encouraged by the results so far. Then there are some species groups that don't appear to be natural. And those generally didn't have very good morphological support anyway, so it's not really too surprising. And so I mentioned before, we're trying to continue to analyze this data to look for additional diagnostic regions for mainly for the pest species. And so one thing we're trying to do is kind of synthesize all of the different types of studies that we've been doing to help resolve some of the problems in Anastrepha taxonomy. One of them is the Furturculus complex, which has been thought to include multiple cryptic species, but it's been really hard to kind of tease them apart and figure out diagnostic characters for them. Our AHE tree shows 6 different lineages, and it confirms that the complex is polyphyletic. Like, different cryptic species don't seem to be necessarily closely related to each other. And 4 of the lineages here, these red ones, were previously recognized, but we think there's perhaps 2 additional ones shown here by the yellow arrows. And then another result was there's been something called the Mexican morphotype of the Furturculus complex, but in our study, this clusters with another species called Zenilde, and there isn't really that much morphological difference there. And I think one problem with previous studies where people weren't looking at Zenilde as they were looking at the complex, they were only looking at the complex. So we think that really is Anastrepha Zenilde. Let's see, okay. Another study we've been doing is within what used to be called Toxicopana. There's a number of similar species, or would have been recognized as species, that have different body color patterns. For example, Blanchard described 5 species from Argentina, based mainly on differences in their general color banding patterns on the abdomen. Like, some of them are predominantly yellow with brown marks, and others are predominantly brown with yellow marks. But people working for land care research in New Zealand were looking for biological control agents of this plant, which has become a weed in New Zealand. And they got samples, or they reared samples from Uruguay that, based on morphology, would be 3 different species, at least by Blanchard's concepts. But I've looked at them more based on the morphology, and there aren't really any significant genitalic differences, and there also seems to be some intergradation of the color patterns. And when we looked at the CO1 sequences, they were almost completely identical. And then if you look at the AHE tree, all of those species came out together. Oh, wait a minute, sorry. This might be the, sorry, this is part of the CO1 tree. This part, oops. this part over here is this part of the tree here. And then in the AHE tree, the same thing happens. Those what are supposed to be different species based on the morphology are all clustered together with no real pattern. So our conclusion is that that's really all one species that has different color morphs, and that a lot of these names should all be synonymized. This group is thought also to be, they're supposed to be thought to be wasp mimics. So perhaps, the reason they have so many different morphs is perhaps in different areas where you have different vespid wasps as models, maybe their color patterns are evolving to match those wasps. So just a quick summary, we did a lot of sampling to try to increase our knowledge of the Anastrepha diversity and distribution and host plants. We have some new molecular tools, diagnostic for most species of Anastrepha. The CO1 data works well for the majority of the species, but for the others, we're trying to look for other diagnostic markers. And then finally, the AHE phylogeny generally corroborates the existing species groups of Anastrepha that have been recognized, and it also resolves many previous questions about relationships within the genes. And that was the end. I hope I haven't put
Thank
you
you
all to
very
sleep.
much. Great. You can applaud, or we can put reactions. This is a Zoom meeting, right? So you can do this. Thumbs up. So we'll take some questions now, and anybody would like to ask, please unmute and ask your questions.
I have a question. Hey,
Hi, George. How are you?
good. How you doing?
All right.
I just out of curiosity, how old is the Tephritidae? Do we know?
My guess would be Eocene. There are some fossils that have showed up in Dominican amber, which is Miocene age.
Okay.
There's been suggestions like during the Eocene there was tropical forest spread all around the world, and so that might have been a opportune time for Tephritids to spread out. There aren't any Gondwanan relationships within the family, so they're not. I don't think any of the Tephritoids are thought to be that old.
Okay. Remind me, how old is the Eocene?
That's what, 40 million years ago, something
Oh,
like that.
okay. Man, they're much older than the Choropids.
Yeah. Oh, we don't really know. Nobody's really done like any. Well, I shouldn't say that. Some people have done, dating based on molecular phylogenies, but I don't know. I always take that with a grain of salt.
Yeah. Tell you one thing, they are a bunch of beautiful flies. Really a pretty group.
Yeah. I feel lucky that way.
Yeah. Thank you.
Sure.
Hey
Anybody
guys,
else? Alan?
I'm gonna take a venture the last mention, because if there's something that the Tephritids are not, is that they're not dull. And this is one question I have to ask Alan, is that what do you think it might be related to that? I mean, we know there's some sexual selection going on, but is there any information that we have about any kind of, do they show any display that they have any special colors for defending from, I don't know, they have special.
Well,
I don't know.
I think it's a complicated situation. I mean, lot of the
Yeah,
related
we-
families are similar, you know, in terms of being.
I
Joey,
came
having-
from Drosophila, and they're very dull. So I mean, if there's something that
Yeah.
Drosophila, so
Well, except the Hawaiian ones.
yeah, except the Hawaiian ones. Yeah, absolutely. But something that fascinates me is that they're not, and I don't know if they show that some display only for sexual selection, or they have some, for instance, they show some distasteful. They have some have anyone tried it where they have any kind of association with taste or something?
Yeah, I don't know if any are really aposematic. Like I don't know of any studies where somebody showed
Yeah,
they're
what about that?
poisonous, but I mean, like a lot of the Anastrepha feed on plants that are considered pretty toxic.
Yeah, but considering the whole group, Anastrepha are probably the dullest of them all.
Maybe, I don't know. Some pretty cool Anastrepha too, but I don't know. I mean, there's different things going on. There's got to be different selective pressures. Like one thing you can look at is the wing pattern, right? So there are certain patterns you see pop up in different genera and different families that look pretty similar. For example, like some of the ones that have banded wings, it's thought that they might be spider mimics, jumping spider mimics, which are some of their main predators. So there's that pressure going on for some of them. Some of them are clearly wasp mimics. But there's a lot of patterns, like within the Tephritidae, you get all these different spotted patterns, and I don't know what the selection might be for there. But there's also probably sexual selection going on. They obviously in courtship, they do all kinds of waving of their wings, so that would make sense. So I don't know. I can't give you any real specific answers there. I think there's a lot of different factors.
So Alan, there was a couple things in the chat, and maybe we could address those, and then of course anybody else who wants to, please feel free. One gentleman has asked whether or not he says he enjoys. It's Paul. I don't know Paul, but Paul asks, as an amateur who enjoys photographing Diptera, what characteristics should I look for that separate Tephritids from other pattern wing flies like the Eulalidae or Ricardidae?
It's probably not all that easy to recognize them in the field. One character, a lot of Eulalidiids and some Richardiids are metallic. Like they tend to be metallic blue. And Tephritids, with very rare exceptions, aren't metallic. So that's one handy field character. But a lot of the characters you need for separating them, you have to look pretty carefully. Like Richardiids always have, well, not all of them, but the majority have a seta on the base of the abdomen. It's kind of an outstanding seta, much bigger than all the other ones. They often have spines on their femora, which Tephritids don't have. Well, actually, there are some Tephritids that do have them, but most don't. I don't know. I've been working on them so long that I just tend to recognize the wing patterns that are common in like what's typical of different genera, and so I'll see the wing pattern and I say, oh, that's genus X, and so therefore it's a Richardiid, or therefore it's a Tephritid. I don't really have any simple way to separate them.
Okay. Another question in the chat box from Dave here was, what % ethanol is recommended for preserving Tephritid specimens?
I recommend 95% ethanol because it's pretty good for preserving the DNA, like better than 70%.
Mhm.
And if you use 100%, it tends to make them brittle and all the CD will fall off. So I usually use 95%. That works pretty well.
I get brittle too with 100% ethanol.
Yeah. Yeah. I mean, if you're preserving them for molecular studies, it's also really good to put them in a freezer right away. Try to keep them cold. It's useful.
I think that's actually called smashed.
Yeah. Right. Yeah.
Thank
You
you.
don't want to put too many specimens in a vial, because if you put too many in, it kind of like the bodily fluids tend to dilute the ethanol down. But anyway, basic answer is 95% is good.
All right. Thank you.
Sure.
Alan, I've got a question for you. I'm curious about those bamboo specialists that you had a photo that they were on the big palm weevil.
Hmm.
Do they use the oviposition or the feeding holes of the palm weevil, and then they have to complete the development and it close in time before the bamboo seals up? Or do they have the ability to get out of the bamboo on their own?
No, they get out somehow on their own. Like the weevil's also feeding on the bamboo. I don't know that the weevils that well, but so they lay their eggs in the inner node of the bamboo, and they feed in there. And then I guess what happens is the bamboo dies and cracks, and then maybe that's how they can get out.
Okay, yeah.
Or unless maybe in that case, maybe the weevils chew their way out and then they can get out.
And provide them an exit hole or something like that. Yeah.
Yeah, I didn't think to ask that question. But there's
Well,
that whole tribe, the Gastrozonini, feed in bamboo, and they do different things. Like some are more like stem miners, in the growing part, but then there's some that are in the internodes, like that one. And they're kind of semi-aquatic, because it's a lot of times there's a lot of water gets into the internodes.
oh yeah, totally. Yeah, I just never heard of that, so that was very cool to learn.
Yeah,
Thanks.
yeah, it is. There's let's see, his name is Damar Kovac, who has been investigating them a lot. He's published some real nice work on those.
Oh,
Um,
cool.
I keep thinking, oh, something's going to turn up in the New World, in the bamboo over here. But there's some eulideids that seem to be in the same kind of niche. Like they seem to be feeding on bamboo. So far, the Tephritidae don't seem to be doing that.
Huh. Interesting.
Yeah.
So
Yeah, they're
I have-
quite nice looking flies too. They're
So Alan, may I ask a quick question also? First of all, I was intrigued by your software that you're clicking on various characters, and you can help identify various species. That's quite a database there, to be able to have, all these characters at your fingertips, be able to click on them and bring up images and all that. That's really nice. I imagine that's a collaboration over a lot of years to assemble something like that. Wanted to ask about when you're doing some of this DNA work. You mentioned transcriptomes. Could you elaborate on transcriptomes? What exactly are we talking about, and how is that used?
to be honest with you, that's probably a question Julian can answer better than I can.
All
But,
right.
yeah, they're involved with RNA. Maybe they actually are RNA.
Yeah, so it's when you're thinking about the genome itself, a very relatively small portion of the genome gets transcribed into proteins that build up all the physical structure that makes everything up. And so the transcriptome portion of it is really looking at the genes that are getting expressed into something that has functional value, rather than the rest of the genome, which includes a whole bunch of non-coding and intronic kind of areas that really don't hold as much evolutionary potential because they are not under the forces of selection. So it's kind of a different way to look at the genomic data that you might be gathering.
So you're looking at introns basically, or rather, you're looking at exons, not introns. You're looking at exons. And when you use the word transcriptomes, is that a collective for all these?
Yeah, generally speaking, the transcriptome is depending on how you are actually pulling that RNA out of whatever tissue you're extracting it from. You're looking at whatever genes are being expressed at a given point of time, whenever that organism got preserved, whenever that fly died and got stuffed in a vial with the preservative, and that's the actual RNA that you're gathering from those kinds of extractions.
Okay, well, that can be quite a bit of genomic material then. In that case, then you have to sort it out and pick out certain gene sequences that are of higher value for doing your evaluations. I'm assuming that's what you'd have to do, right?
Yeah, that's genomics. That's the big issue now. You get so much data, it's overwhelming.
I can
Analyzing
imagine.
it all is really the challenge.
Right. Well, you
I don't know,
said
I think with the
using-
transcriptomes, that was kind of a hot method for a little while, but the methods for getting a broad range of data have improved so much that I don't know if people are using the transcriptomes as much, because it's harder to get samples. You gotta put them in a special fluid to preserve the transcriptomes. So that's not that easy to do. A lot of the other methods are working on just stuff preserved in alcohol, or a lot of them are working pretty well on pinned, dried pin stuff. It's not too
the
old.
CO1, for example, that you mentioned, right?
Yeah, the CO1 is usually pretty easy to get because it's a mitochondrial gene. So there's lots and lots of copies of it. So that's usually one of the easier things to get. Sometimes you can get that out of, not so well preserved material that you can't get, full genomic data.
And of course, over time, the mitochondrial DNA is something that's conserved, if I'm not mistaken, right?
Yeah, I think it depends on a lot of factors, how the material is kept, and what preservation method is used, and things like that.
Anybody else would like to ask questions?
I want to dovetail off a comment that Fred made. He was talking about how much he liked your matrix-based key, and I have seen Jason Dabrowski's matrix-based key for Canadian Lepidoptera, and I fell in love with it. I thought, wow, that's really where I'd love to see us go eventually. Alan, are the matrix-based keys becoming more popular, more common?
Yeah, I think for a lot of pest groups now, there's keys available. USDA APHIS had a group that was trying to fund development of keys like that for pest groups. So there's quite a bit available.
Are they publicly available, or usually just for the APHIS community?
No, they're publicly available. That APHIS group had a website. I don't know if I can remember the name of it, but they had basically a couple of pages of links to different keys for different groups. Like I say, they're mostly for pest insects, but they're really useful. Like, I mean, for a small genus, you know, you don't really need that. You know, if there's 4 or 5 species, it's not really worth developing that kind of key. But where it's really useful is for really big ones, where you just can't, you know, keep track of the information. Like in a traditional kind of publication. So by using them, you can kind of put all the information into, you know, one source. So you got all your, you know, illustrations and all your diagnostic information.
Yeah, what I liked about the most is you sometimes only had to enter maybe 2, 3, or 4 morphologic characteristics, and you could pin it down to 1 or just a small number of species.
Yeah. You know, you still kind of need to learn what are the best characters to use.
Right.
I mean, you can follow what the key recommends, but it really helps to be familiar with those. But yeah, I mean, I use the key myself all the time.
Imagine you would.
Yeah, like, you know, like we were getting, you know, tons of material in from some of these sites, so, you know, I can't keep it all in my head when you have that many species. So.
It's a huge amount of information. It's overwhelming, really.
Yeah.
Are there any Tephritids in this country that might have been considered or are being used even for weed biocontrol?
Yeah, there's like 10, you mean, that are from here that have been used elsewhere, or
Either
brought
way.
in?
Yeah, either way, just curious. Either
Yeah,
way.
there's like 10 species that have been brought in, mainly for thistles and
All
other
right.
rangeland weeds. Yeah, there's a couple you can find around here. They're on knapweeds. I don't know if you know what knapweeds are. They're kind of purple flowered. You find them on the roadsides and stuff.
Yeah, they can overwhelm an area easily.
Yep. You know, they've kind of had mixed success. Like, I don't think any Tephritids have like completely, you know, been the silver bullet to control a weed, but
Right.
some of them are thought to, you know, they reduce the ones that feed on the seed heads, you know, reduce the amount of seeds that plants can produce.
Yeah.
And so, you know, they've been considered worthwhile, I think, to, you know, to bring in. Yeah, and I mentioned that Anastrepha, like people in New Zealand are studying that as a possible biocontrol agent. There.
Do we get that weed in our southern borders?
Trying to think if we have it. I think it might be in Florida as well.
Yeah, not surprising, Florida. But
Yeah, they get a lot of things.
yeah, I haven't seen it. I was looking at the image you had of the plant and I didn't recognize it. But
Yeah, I don't know it either. The fly actually hits a variety of it's in Asclepiad, and it's got like a big it's a fruit with a big thick husk. So this is in the group where the female ovipositor is really long, and the larvae develop on the seeds that are inside. So the female needs a long ovipositor to get the eggs all the way through the husk into the internal part. But that species is known to feed on a few different Asclepiads, so it's not super specific, but at least in New Zealand, I think they only have the one weed, so they're not really as concerned about it jumping to something else.
right.
Bringing it into the US might be more difficult. If there's anything else related, it's already here, it's native.
Well, we do have native Asclepiads. I don't know if that would be any threat to them, though, necessarily. But
Yeah, I mean, that'll have to be evaluated.
Yeah,
if it's done right.
exactly.
Right.
Okay. Anybody else would like to ask? Let me check the chat box in case there's anything further there. Nope.
Yeah, I'll ask Alan.
Mhm.
Do you expect to start working on a Princeton field guide for the Northeast? Something like Skevington and Locke could put out.
I'm not a very good photographer, so I probably couldn't match what Jeff did. That's a really nice book. There is a handbook. I helped Dick Foote out with, helped out Dick Foote and Louis Blanc. That was kind of the summary of their careers. It came out in 1993, so it works pretty well for it's for the U.S. and Canada.
Okay.
If I was going to do anything, it might be to try to update that. I know Gary Steck and Bruce Sutton and I were. It's mainly those two. I've been working in the Smoky Mountains. They were talking about putting out a sort of a guide for that region, and that would cover a lot of the eastern U.S. But it's not imminent.
We'll look forward to it down the road.
Yeah, but Gary's a good photographer. That would have good images in it.
All right, well, in the absence of any other questions, then I guess I want to thank Alan once again for a terrific presentation and a lot of really great information, especially for us who aren't familiar with this taxa at all. So we appreciate you lecturing for us tonight, Alan. Thank you very much, and look forward to seeing you somewhere down the road at the museum one day or something. So you are an honorary member of MES now.
Oh, okay, thanks.
Sure, it's us who thank you, and Jean will have a journal sent to you as a courtesy also. And I guess I could email you and get your contact mailing information so we can have that sent. Okay.
Oh, sure.
Yeah, I'll contact Alan. I'll send him an
Okay.
email tomorrow, and then we
Great.
can work out the details.
Okay. So, Julian, thank you also for your explanation. I appreciate that. And thank you, everybody, for joining us tonight. Good to see you. Dave, take care, man. And next month we're meeting, we said already, I'm sorry, we did say we're going to do a hybrid meeting. So I hope that'll all work out. So, but thank you again, folks, and everybody have a great weekend.
All right, you too, Fred. Ronaldo,
Okay,
I'll
thank
look forward to see you next week. We're actually meeting up in Kentucky next week.
Yep, looking forward to.
All
Good
right.
to see you, Alan. Great presentation
Great
as
seeing you.
well.
Oh, hi,
Hey,
Dan.
we can see your face. Bye-bye.
All right.
Thanks
This
all.
internet now sucks.
All right. Well, good night, everybody.
Good night.
night.
Good night,
Thanks
Alan.
again.
Thank
Good
you.
seeing you,
Bye.
Gene.
See you, Emil.
Yep. Bye.